This method provides an original solution for cellular analysis, a field that is currently lacking suitable recording systems that are multiparametric, biocompatible, mechanically compliant, and finally low cost.
With this method, it is possible to obtain sensing systems with different sensing capabilities, such as the ability to monitor both the electrical and the metabolic activity of cells using a single type of electronic organic device.
Besides helping to reduce in vivo animal experimentation, in vitro multiparametric systems are an extremely promising tool in a variety of biomedical fields, such as personalized medicine and studies on neurodegenerative diseases.
Owing the high versatility of the organic charge-modulated field effect transistor, it is also possible to integrate other sensors, such as, for example, sensors for the detection of ions of different biomarkers.
Cut 6 by 6 square centimeter pieces from a new 250 micrometer PET sheet. Then, rinse the PET substrates with acetone, isopropyl alcohol, and deionized water. Dry them using streams of nitrogen and store them in clean plastic Petri dishes. For titanium deposition, pre-clean the substrates with plasma oxygen and place them on the substrate holder inside the vacuum chamber of the thermal evaporator. Next, place 60 milligrams of titanium in the crucible, close the shutter, and pump down the evaporation chamber until it reaches 10 to the minus 6 torr. Increase the power of the evaporator until the crucible glows red and wait for 30 seconds.